Hello, everyone. Thank you for taking the time to join us today for this exciting webinar being brought to you by Scintica. My name is Gabriel Escalante, and I will be your host for today's webinar. The webinar today is entitled Virtual Demonstration of the IVM System. Today we'll have two presenters, Dr. Selina Ann, Application Specialist at IVM Technology, and Dr. Nilofar Khosravi, Product Manager within the Imaging Division at Scintica. Allow me to provide some background information about our guest speakers. Dr. Selina Ann has been working in the intravital imaging research field since 2014. She received a PhD in the Graduate School of Nanoscience and Technology from the Korea Advanced Institute of Science and Technology in 2020. Dr. Ann has focused her research on understanding in vivo multi-phase spatial temporal cellular dynamics of transplanted HSPC in bone marrow during early engraftment using a custom-designed video rate intravital confocal microscopy system. In addition, she has extensive in vivo research experience on comprehensive 3D spatial temporal visualization and imaging analysis of various organs such as lymph node, lung, liver, spleen, and brain. As an application specialist in IVM technology, she provides hands-on practical support for researchers in various research fields to take full advantage of intravital imaging techniques to maximize their research performance. Nilofar is a transnational medical scientist specialized in regenerative medicine and molecular imaging. She holds a PhD from the University of Toronto and a Master's in Science in Biomedical Engineering from McGill University. Her work has been centered around developing novel technologies for the minimally invasive diagnostics and therapeutics. She has extensive experience in designing and developing platforms for intravital imaging as well as post-acquisition analysis. Nilofar also serves as an advisor for the Academy of Osseointegration and has a successful track record in obtaining national and international awards. She is the author and inventor of high-impact publications and patents in the fields of molecular imaging, implantology, regenerative medicine, and wound care. During this webinar, we will provide you with molecular imaging methods in brief, examples of intravital microscopy used for real-time visualization of live tissue organs. Additionally, we will also demonstrate the IVM-MS, a new rapid all-in-one technology for intravital microscopy. Finally, we'll finish with animal models and applications examples of the technique. After this webinar, you will understand the workflow of the IVM system, its hardware and software components, and its capabilities to acquire quantifiable microscopic images from dynamic processes occurring in live animals. As some of you may know, Scintica is the distributor of IVM's intravital microscopy platform systems in North America. Today's webinar is the final session in a 4-part series, so if you're interested in viewing the previous session, please visit our website. Before we begin, I'd like to briefly mention a few housekeeping rules. We anticipate that today's presentation will be 45 minutes, so we'll have time to answer your questions live. Please feel free to submit your questions to the Q&A dialog box throughout the presentation. Additionally, we'll create a transcript of the questions and we'll distribute this document within the next week or so. Please also note that we're recording today's session, so if for some reason you lose the connection or cannot hear everything clearly, you will have an access to the video file to review later. Without further ado, I'm going to pass things over to Nilofar and Dr. Ann.
Thank you so much for the introduction, Gabrielle. Hello, everyone. Thanks for attending our virtual demo of the IVM system today. My name is Nilofar Khosravi, and I'm going to get you started by a short introduction to the intravital microscopy system and its components. To refresh your memory on the system and give you a little bit of background to those of you who missed our previous webinar sessions. Then I'm going to pass it over to my co-host, Dr. Selina, and she will show you the procedure for preparation of the animals and then imaging of various external and internal organs of the body. So IVM system is an all-in-one intravital, confocal, and two-photon microscopy system, which is designed and optimized for imaging of live animals. So those of you who are familiar with the world of intravital microscopy would know how using conventional confocal microscopes for animal studies can be technically challenging, as you as users have to prepare every single component and function of it by yourself. So the main challenges and limitations lie within the depth of imaging and also motion artifacts due to lack of proper stabilization of the animal, and also some of the physiological activities such as breathing and the brain pulse will cause artifacts to your images. And ultimately, taking care of the animal welfare and the temperature control is kind of challenging with the conventional confocal microscopes. IVM Technologies has come up with a solution to make intravital microscopy easier and reproducible for non-expert as well as for experts. This all-in-one single box package enables easy installation, operation, and maintenance. And it comes with a hardware and software that are optimized for ultra-fast imaging, and which offers no limitation in imaging various internal organs. So this system is equipped with live motion compensation function. So it helps us get the high quality images of moving organs. And also it comes with specific hardware that has been integrated in the system to maintain animal normal physiological conditions. I'm going to touch upon all these functions and elements throughout the presentation, and we're going to show all those. There are 4 different configurations for this system. It comes as a confocal only system or a 2-photon only system. And we also have one which has a combined confocal and 2-photon laser. And the newest version is a smart system, which is a 2-photon smart system having a laser fixed at 920 nanometer wavelength for deep tissue imaging. So very quickly, I'm going to remind you about this microscopy techniques. What is a confocal and what is a 2-photon? If you don't have any background, then you never used it before. Confocal microscopy is an optical imaging technique that uses a pinhole to block out of focus light coming from the background tissue. So it increases optical resolution and the contrast of the images acquired. As you see here in the examples, it has a great contrast between the tissue and the background. So it basically offers you this capability to optically section the tissue as you go deep through the tissue. So that's why it gives you a high resolution. 2-photon microscopy, on the other hand, is a technique that utilizes 2 photons of a longer wavelengths instead of 1 photon, but these 2 photons have a lower energy to excite fluorophores. So as you might know, components of the tissue, such as hemoglobin, absorb and scatter incident excitation light of most visible wavelengths. So this would limit light penetration, and it would result in low emission signals that are difficult to detect consistently. The 2-photon technique uses near infrared excitation light, and since tissues are effectively, a lot of them transparent at these wavelengths, the background signal is strongly suppressed. So the combination of these effects that I just explained increases the penetration depth and reduces the photo bleaching of your sample. So that's why often for deep tissue microscopy, 2-photon is used instead of a confocal. And also a great feature of it is that with a 2-photon laser, chameleon laser, you can, it gives you the ability for label-free imaging via second harmonic generation. So this second harmonic generation is usually used to look at fibrous tissue, as it shows fibers very well. So fibrous tissue such as bone and muscle. And this example, we have imaged muscle. You will see the sarcomere visualized by second harmonic generation, and vessels are in CD31 blue, and then you will see the neuromuscular junction in yellow. Now that you have a little bit of background about the techniques, I'm gonna talk to you about the components of the system. So this is like other microscopy units. This comes with a microscope body, the black and white box, and there's underneath the 2-photon laser box, and there is a system power supply. Of course, it comes with a computer and a screen to monitor. And then there is a stage controller to control the motorized stage. And there's the heating pad controller for animal physiological control. And there are some accessories associated with this system that it's unique to this instrument. This is this body temperature control system that you will see on the top left-hand side. And then we have an inhalation anesthesia set, which is custom designed for this instrument. If you have used anesthetic agents with in conjunction with window chambers in the past, you would know that it's hard to fit the nose cone and the window chamber all together. So we have optimized all these equipments to be going well with the window chambers. And then we have these imaging window chambers and the holders you see at the bottom left-hand side. This is an example of thoracic window chamber model. And on the left, we'll have a stereotactic mount for brain imaging to stabilize the head. As I mentioned briefly before, the animal maintenance platforms that this instrument comes with, it has a plate heater that can maintain body temperature. It is connected to a rectal probe, which sends feedback to the system, and the temperature is automatically adjusted. If you need to move an organ slightly out of the body for imaging, then there is a tissue temperature heater with integrated grass cover slip that comes in contact to the tissue and kind of plays the same role as the rectal probe, and it sends signals and automatically controls and maintain the temperature. So you need to keep in mind that if you're imaging a number of animals, so you need to change this cover slip when switching from one animal to the other to control, to stop the contamination. So another cool feature of the system is the design of it, which is designed as a closed box with a sliding blackout door. So it protects your experimental sample from ambient light. So unlike other microscopes that you needed to be placing them in a dark rooms with curtains running from ceiling to the floor. So this one can be placed in any room with any lighting source that you desire to have in your room. So without needing to worry about the light penetrating your chamber. So now I'm going to briefly mention about the window chambers, and I'm going to go back to it later on through the webinar. If you are planning to perform longitudinal studies, you are required to place window chamber in your animals to A, to optically expose the organ or tissue of your interest, B, to preserve the area from all sort of infections, because when you go put back your animals to the cage to recover from the, let's say, anesthesia, then you need to protect the area which was exposed for the experiment, and you can keep the animal healthy for your next time point. So that's the role basically the window chambers are playing. And this allows you to track one animal over time, which reduces the variation between animal to animal in your data. And also it reduces the number of animals used in your cohort, which is ethically very important. So we have collected a series of videos to show you examples of imaging procedure with or without window chambers. So we were planning to do this demo live in the lab and show you as we were doing all these experiments, but due to ethical concerns, we were unable to video live in the lab. So what we tried our best to bring these videos to you in a way that you see all the steps clearly. So in these videos, we will walk you through the procedure and show you how those experiments work and what type of results you can expect from this type of experiments. For today, we have picked a few models to demonstrate. First, we're going to start by showing organ models. The first model is an external organ model, which is ear skin. It's very minimally invasive. You basically need to do a very little preparation before you can get images. And then we're going to move towards imaging internal organs such as spleen and lung. And then at the end, I'm going to show you some videos of the procedure for placement of cranial, dorsal, and thoracic window chamber models and some of the images that we collected using these models. So before we get into the lab area and start to do the imaging, we need to start up the system. So this is like any other instrument or microscope. Of course, you need to switch the machine on. And typically before you switch the entire power box and everything else or pull up the software, we need to switch on the laser. So the reason is we need the laser to be warmed up for 5 minutes before we turn on everything else. This is really important. And then we switch on the machine itself, and then there is a software that we click on. It's called IVM engine, and we give it a few seconds for the program to boot. So I'm going to quickly walk you through the graphical user interface of the software and some of the function that the software does before Selena going to show you everything in detail. So there's a capture tab where you control the capturing of your images, like videos and all those sorts of controllers. And there's a control tab, which is very crucial for you to set up your experiments. You need to set your two-photon laser power and the detector for operation. And there is a Z stack function which sets you up automated imaging along the Z stack. If you are trying to image a thick section, you need to set up a Z stack to go through the tissue. And then there is a mosaic function if you're setting up an automated image along the X and Y plane to increase your field view. And then you need to tile these various images that you acquire. Let's say if you want to image a 2 by 2 millimeter area, then you need to collect, let's say 6 images and tile them together. So that's the role of the mosaic function. And then there is time lapse, which gives you ability to image over time at preset time intervals. Let's say I can set for the course of 10 minutes, take one image out of my ROI at every 20 seconds. So that's it helps you to correct the series of images, which you can later turn it on into video. And there is a multi-position tab, which allows you to take an image of multiple positions, random position around your tissue. So it automatically does this imaging for you, the software. And you also have ability to combine all these functions. Let's say you can do time lapse and multi-position and tile. So it's all possible. It depends on your experiment. And at the end, there is an IVM studio, which is prepared for processing and analysis of your images. So to talk about a little more about the controller tab. This is to set and adjust the laser power. Let's talk about the laser power for a bit. It really depends on the tissue you want to look at. You choose your laser power. I would say, to give you an idea of the number, I would say normally it's set around 20%, but it really depends on how deep you want to go in the tissue. and what you want to look at. So when we use a high power, let's say to look at tumor, because tumor is dense, and if you want to look at the mass score, you need to crank up the laser. So this is the same story for deep tissue imaging. You need to increase the laser to get more signal. The detector, on the other hand, is your gain. So if you, of course, increase the gain, you will receive more signals, but from your area of imaging. But of course, you're gonna also get more background. So it's important to find the right balance between the power and the gain for every channel that you're imaging to get the best contrast. So after it's pretty straightforward. You set the folder and the directory for the files you're gonna save. You select your objective lens. So it depends on what lenses you're gonna physically place into your configuration setup. So it can go as low as 4x or as high as 100x or 60x. So, and some of these lasers are dry, or some of them are in water or oil immersion to increase the resolution. And then there is the size of the image that you can adjust. So it's usually it's 512 by 512 pixels, but of course you can go higher, 1024 by 1024, and you need to keep in mind, if you increase the size of the image, and you're gonna be also increasing the scanning time, which means that it will be more time that your laser interacts with your tissue and increases the photo bleaching. So next, before we acquire the images, we need to select our channels. So if there's multiple like fluorescent channels, green, red, yellow, far-red, that we can image with the system. There's whole spectrum range. But let's say you can also assign pseudo colors to your channels, but typically people assign green to GFP, red to RFP, but this image is gonna at the end come black and white, and you're free to choose your colors in these images. When we set all these components, I would say these are the basic components that I walked you through. So at this stage, the system will be ready to acquire images or record videos. So at this point, I'm gonna passing it over to Selena to show you the procedure for preparation of the animals and acquisition of the images.
Thank you, Nilüfer, for the introduction. Hi, everyone. I'm Selena from IVIM Technology. Here I'll talk about the experimental procedures of the intravital imaging of various organs using IVM system. Let me introduce how to do the in vivo imaging of ear skin. For intravital imaging, gently grab the mouse. For the injection like this, the mouse was anesthetized by IP injection of ketamine and xylazine. Before imaging, blood vessels were fluorescently labeled by intravenous injection of anti-CD31 monoclonal antibody, which is conjugated with a fluorophore, or various fluorescent dyes such as FITC dextran, TriTC, or rhodamine B was injected via tail vein for the in vivo blood vessel labeling. Here, TriTC dye was intravenously injected to the mouse model. Then the imaging area of ear skin was shaved with the hair remover cream for preventing visualization of high autofluorescence signal expressing from the hair. Then we applied the cream evenly to fully cover the hair on ear skin. Then after a few minutes later, gently remove the cream and clean it with saline solution. Then the mouse model is mounted on IVM stage with heating plate functions, and apply cell line on the surface of ear skin imaging area. then cover with cover slip, then place the ear in the middle of cover glass, as shown in the movie, for preventing air bubbles to be captured in the imaging field. Then make the imaging tissue fully attached to the cover slip. After that, drop the distilled water on cover slip for immersing objective lens. With a rectal probe for sensing the body core temperature, which has a feedback system with the stage heating plate, the mouse body temperature is maintained at 36 Celsius degree during the whole imaging time. Then place the imaging area in the middle of objective using stage controller, X, Y, and Z axis. Then adjust the focal distance by minimizing the distance between specimen and objective lens using a stage dock, and close the door for preventing light from outside. When we look up the imaging software, actually this model is IVM-C-M. Both Confocal and Two-Photon microscope are combined in one system. And this time we select the Confocal mode here and then start playing scanner for acquiring imaging with 1024 pixels each in X and Y. And turn on the laser power of each channel and detectors. And then just control the detector gain with master controller. Then set the averaging rate as 1. For setting the directory file to save the acquired data, click Set Folder and set the directory file. And in the note, we can set file name to be created following the numbering. And then later shutter on for initiating the excitation. And then lower the stage in Z-axis using stage controller until we find the focal signal. In a live window, we can see the real-time fast dynamic movie. Here's the raw movie data of blood dynamics in ear skin. We can see the fast dynamics of RBC flowing in the blood vasculatures. When we capture this, we can get the result image on the image display window in the right. After finding another interest imaging area, capture the image, and here comes the result raw image file. And when we record the real-time video, then this is the real-time movie of blood flow in the blood vessel in ear skin. After that, if we click Open Folder, then we could find every raw data we acquired in the directory file. Actually, for the image data we captured is created as PNG file, and in case of recorded movie data, we'll come up with the AVI file. And for acquiring the clear data from the moving animal by checking motion compensation function, then select the channel for the registration reference, which is normally includes some non-moving object. Then after capturing, we can get the compensated clear image on the right. For the compensation, this is the result with and without motion compensation function. The image without motion compensation looks very shifted with motion. But while after compensation, we can get very fine and clear images in the right. with IVM Studio, the image processing software, we can process the data after data acquiring using engine software. Then open the folder we got. We can see the movie and the image data here. And also we can control the brightness and the contrast of the data on the color tab, and can crop or make an annotation in image tab too. And also in image processing tab, we can make the data which is acquired with imaging mode, such as Z-stack, time-lapse, and mosaic functions to be registered along Z-axis or time frame, and make a projection and also tiling image in this tab. And every image modes automatically created with their own files, and each mode tab is activated accordingly. And for intravital imaging of spleen, the skin near the spleen was firstly shaped with a hair clipper to trim the long hairs on the skin for applying the hair remover cream. And then apply the declaratory cream to fully cover the hair for a complete hair remover. After a few minutes, gently remove the cream with a dry ear tips, and then clean with saline and alcohol swab. The imaging target tissue, the spleen, was exposed with the skin incision using various surgical tools, such as tweezers and surgical scissors, and then widely open the skin to fully attack, to fully approach to the imaging organ, and then gently take it out using ear tips and place the wet gauze under the imaging organ. Then the mouse model is mounted on IVM stage with heating plate functions. Then apply saline on the surface of imaging area using ear tips. Then place the specimen in the center of the stage and cover with cover slip. And drop the distilled water on cover slip for immersing objective lens. Insert the rectal probe, which is optimized for tissue temperature control module. Then adjust the focal distance between the imaging sample and objective using stage controller. And close the both side of doors for preventing light from outside. And this is the result of in vivo imaging of spleen and pancreas. We could visualize real-time cell dynamics of the endogenous monocyte and dendritic cells in spleen by using CX3CR1 GFP transgenic mouse. And this is the example of pancreatic islet cells imaging using MIP GFP transgenic mouse. Those islet beta cells in pancreas were visualized in vivo, and their cell dynamics and the development of islet cells were monitored in long-term basis by using the intravital abdominal imaging window. As you can see here, we can get the real-time cell dynamics of islet beta cells in pancreas, in spleen, sorry. For intravital imaging of liver, the abdominal skin near the liver was shaped with a hair clipper too. And then apply declaratory cream again to fully cover the hair. After a few minutes later, gently remove the cream with ear tips and then clean with wet gauze with saline to remove the cream fully on the skin. The imaging tissue, liver, was exposed with the skin incision. So incise the skin wide enough to easily approach to the target organ, which is liver. Yeah. Incise it widely enough, and then gently take the organ tissue slightly out of the body using air tips and place the wet gauze under the imaging organ. The mouse model is mounted on IVM stage with a heating plate and slightly covered with cover slip. And this is the example of the nanoparticle delivery and accumulation in the liver sinusoid, capturing by real-time video. The red fluorescent nanoparticle were delivered in liver sinusoid right after the injection via tailbane catheter, and then accumulated to the specific size of the liver sinusoid a long time. You can see the dynamics of the nanoparticle accumulations to the specific size. When we apply this live in vivo liver imaging technique to the fatty liver disease mouse model and liver fibrosis model, we can clearly visualize the development of the fibrotic collagen and the lipid drumlet in liver tissue. And for the experimental method of window imaging chamber mouse model for longitudinal in vivo imaging, I'm going to pass over the presentation to the Niloufar.
Thank you, Selena, for showing us the procedure. So I'm gonna now walk you through the procedure for placement of window chambers as I promised. So now you observed the basic steps that you take through the imaging process to look at organ models, different various organ models. I'm now going to quickly show you examples of placing window chambers for, as I mentioned, sake of longitudinal imaging. I'm going to start with the cranial window chamber model, which is usually used to look at subcranial tissues such as dura mater and brain. So here you see the procedure pretty similar when the animal is intubated, and then you will make an incision into the skin and the clear area out of periosteum, and you use a high-speed drill with a terifin attached at various diameters that you want to cut. You loosen the bone, and then with a forceps or tweezer, you flap the bone out. And then you use a dental restorative material to cover the area, basically to secure a cover slip in place. So this material is used clinically, so there's no harm to the animal. And then you use a light curing unit to polymerize and stabilize this adhesive. So you make sure that your cover slip is attached to the skull. And then you mount the animal on the stereotactic mount designed for cranial, and you put it under the microscope for imaging. So this is the example of what you will see through a cranial window chamber model. And so this is imaging of blood-brain barrier model and the Alzheimer's model. So you will see here on the image on the left-hand side, nicely endothelial cells are lining up, which are labeled with CD31 in blue, and then there are pericytes, those cells that are wrapping around the vessels. Those are mesenchymal cells that they have regeneration capabilities. So they are labeled with a red fluorescent protein, but the reason that you see them purple in this image is because of the overlap between the two channels of red and blue, as these vessels are meant to be tightly bound to the vessels, sharing a membrane with endothelial cells. And there are astrocytes that are labeled with YFP. So this type of images allows analysis of vasculature in 3D, and you can track the changes to the morphology of the vessels over time as you're imaging through the cranial window chamber. So this is an example of an optically cleared brain tissue. So it clearly shows that how deep you can go with two-photon imaging. It can go up to 1 millimeter if you look at the scale. So for brain is one of those organs that you can go really deep into, as it's not fibrous. And here you will see the neural cells that are labeled with YFP. So now the next procedure is a placement of the dorsal skin window chamber. So we have the hardware custom designed for this window chambers. You place the substrates around the skin, and then you tighten up with screws to fix it in place here. And then to make sure it's tight sealed, you can suture around this substrates. Now you make sure there's no fluid coming in or going out, and the skin on one side of the window chamber can be just thinned, so you can get a clear, like a better resolution. And also on the other side of the chamber, you put water on top to increase the resolution, and then in the other side you can inject tumor cells to inoculate your samples for cancer models, and you place car slip on top of the area to preserve it. And this is the ring you put it to secure the car slip in place. So to ensure that no agents can penetrate into the imaging area, and there will be no artifacts. So now you place it under the microscope on a microscope stage, and you go through your common imaging procedure. So you can, you know that this is safe to put this animal back to the cage and bring it back for your next imaging time point. So this is an example of what you would see through the dorsal skin window chamber. And this is monitoring of nanoparticle delivery to triple negative breast cancer. So a drug is delivered via nanoparticles to the tumor cells here, which are labeled with GFP. And the nanoparticles, which are really small here, if you want to look at it, you need to magnify, but you can still see them in red in these images. And there are vessels which are labeled with CD31 in blue. So you will see that this is a longitudinal imaging, so which is captured 2 hours, 6 hours, and 24 hours post injection of these nano particles intravenously. And you can monitor the progression and regression of your tumors over time, and also the effect of these therapeutic agents. So the next window chamber model that I'm going to show is the thoracic window chamber model to image the moving organs such as lung and cardiac tissue. So the procedure is pretty similar to the others. To start with, you intubate your animal, and you surgically expose a lung or cardiac by creating an incision into the skin. and you place, this is the look of the thoracic window chamber. As you see here, there is an airway associated with this chamber, which is custom designed, and it's connected to a vacuum pump. So when you turn on the vacuum pump, it allows to sucks the air and allows a small portion of your liver or cardiac to be coming closer to the curve slip. So you can stabilize that with a vacuum-based method, and you can image that area. But still, it's gonna be a moving organ. If we don't have this motion compensation function, you won't be able to see these tissues clearly. So you see the example image taken through this window chamber from lung, and there are vessels that are showing up in green, and there are circulating neutrophils that are in red. So the quality is really good for a moving objects such as lung. And now this is an imaging of cardiac tissue, another model that we can use the thoracic window chamber to look at. You see how dynamic is this tissue moving. This is the live scanning, which you see the beating heart here. And with the motion compensation function, we can get a clear, non-blurred image of this tissue. So you will see here the lymphatic vessels that are labeled in GFP, and there are blood vessels that are labeled in red, and there are just different in sizes that you will clearly see the capillaries versus lymphatic vessels in the heart. Now I want to end today's demo presentation with this beautiful slide, which shows you how we are capable of imaging almost every organ or tissue that you can imagine in animal's body. So you can look here at the adipose tissue, skeletal muscle, lung, ear, skin that we already show you, lymph nodes, retina, small intestine, kidney, and so many others. So you just need to design your experiment and come up with your research question. This instrument will help you look at every organ or tissue at the single cell level resolution. So thank you everyone for attending today's session. Now I'm going to hand it over to Gabrielle to moderate the question and answer session.
Thank you, Dr. Ann and Nilüfer, for the great discussion today. We'll now move on to the live Q&A section. We have a couple of questions, and the first one is, does the IVIM system have a resonant scanner?
I'll answer that question. Actually, as our system is using polygon scanner system, while the microscope from other company using resonant or galvanometer sets. So thereby the default imaging quality is quite better than any other company's system, more fast and better image quality with equivalent illumination of the imaging field. In addition, the real-time averaging recording such as D-mode and motion compensation function and denoise function altogether help users to acquire the high quality data in real-time frame rate.
Thank you very much, Selena. The second question is, for vascular imaging, would you use Confocal or Two-Photon laser?
Oh, we have both of them. We got a confocal system and multi-photon system, and the combined version of the confocal and two-photon microscope system. So all of them are very real fast dynamics we have.
So I need to add one more point that both single and two-photon imaging are capable of capturing the vessel structure. So it really depends on how deep the vasculature is in your tissue. So if you want to really go deep, you need to use the two-photon laser. And the ones that are more superficial can be captured with confocal, but both gives you like a high resolution for vascular imaging.
Thank you very much for the answer. The next question is, in your motion compensation software, what is the what is it different from rigid and non-rigid registration?
Actually, I didn't understand what is rigid or non-rigid registration. Oh, now we got 30 to 6 frames per second video rate. And okay, could you explain what is the rigid and non-rigid registration?
So I'm guessing we're going to get in touch with this attendee so that we can clarify all the questions that he has and give him proper answers.
Okay, very good. Yeah.
For now, we're just going to move on for the next question, which is, what's the imaging time window after injection of vascular dyes?
Okay, for vascular dyes, it usually, let's say for a healthy animal which has proper blood circulation, like vascular circulation, no leaky vessels, no tumor, no injured area. The time window that I can have on top of my head, it's let's say the dye can stay in the vessels for up to 30 minutes after intravenous injection. But also it depends on the size of your agent, labeled agents. For example, we have different molecular weight dextrans. So if you need to look at the leakiness of the vessels, as people usually used to look at tumor vessels, then you need to inject a small molecular weight dye so they can leak out of the vessels. But if you, for normal vascular imaging, if you use a high molecular weight dextran, for example, it can stay in the vessels properly with no leaking, good contrast for up to at least 30 minutes, I would say.
Thank you for the great answer. The next question is, would this system be capable of being incorporated or built into a biosafety cabinet or downdraft table for live imaging or infectious diseases?
So the size of the system is slightly like the microscope itself can fit under a biosafety cabinet. But I assume if there are channels that can be connected directly from the biosafety cabinet, just moving the sample from the BSC to the system, as the good feature of it is that's a closed system. If you want to keep the inside sterile, just wipe it with disinfecting agent. It can be clean, and then you can put your samples and then put it back under the biosafety cabinets. That's the way I can think of. Otherwise, this is the instrument is not small. You cannot put it under a BSC.
Thank you, Nilüfer. We're going to move on to the next question, which is, which objective lens is good enough to look at cells?
Okay, yeah, it really depends on the size of the cells you want to look at. The cells have a very wide range of sizes. For example, I can think of mesenchymal cells being a bit large. So with the 10x objective, you can look at these cells. Like you can look at any cells kind of with a 10x objective, but you won't be able to see the morphology of the cells. If you move towards the 20x, 20x is good enough, let's say, to look at the very small vessels, such as red blood cells that are, I think, in the range of 7 to 5 microns. And if you use any magnification, like above 20x, they're of course more than good enough to look at cells.
Thank you. The next question is, what window models are available to look at bone?
Yeah, for bone, like the cranial one that we just show is a perfect model to look at a bone in the cranial site. So it depends on what function you want to look at in the bone. If you are looking at for non-load bearing bone, of course, Cranial is a great model. If you want to look at the load bearing bone, and there is a model for femur window chambers that you can place it in the femur side, and you can look at expose the bone marrow. You can look at both cortical and cancellous bone with that model. You can do the same with the cranial. Cranial is just, I don't know what's the source of this question, but Cranial also has a bone marrow. It's just a very little layer of bone marrow. It depends on what they want to track, but there are multiple models. I would say Cranial and femur would be great models to look at bone.
Thank you again, Nilufar. The next question is, are you able to see an image blood flow?
Of course, we can see the blood flow because our system is very fast. The frame rate is about 30 to 6 frames per second, which means within 1 second we can gather 30 to 6 frames at the same time. So we can visualize the blood flow and also can perform some in vivo analysis of the blood flow velocity. Yeah.
Thank you, Selena. We have time for one more question, so we're just gonna move in, and it is, which software can be used to analyze the images?
Okay, so the images that you get from the system, that you can analyze them with any software. Of course, IVM Studio is the best you can use because it's easy. That comes with a system. But you can also analyze it with any other software that you are comfortable with. All the open source software such as ImageJ, or the ones that you need to acquire license to use, such as Imaris, if you want to do 3D analysis. But basically any software, and the size of these images in terms of volume, it's very small, like I would say around 700 kilobytes. So you can easily handle it with any computer that you have.
Thank you, Nilufar. We have reached the end of our session for today, and to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of the webinar, we'll be sure to answer any questions in the written transcript, and we'll work to get this out to you over the next week or so. I would like to thank Dr. Ann and Nilufar for the wonderful presentation today, and I trust that we have been able to provide you with some relevant information about the virtual demonstration of the IVM system. And as I mentioned at the beginning of today's webinar, this was the final session of a 4-part series, and if you wish to watch the previous sessions, please visit our website. If in the days and weeks to come you have further questions about the modalities discussed today, I encourage you to reach out to us here at Scintica, and we'll be happy to discuss further. We would welcome the opportunity to discuss your specific research goals and how any of our imaging systems could help move your work forward. Thank you again to all of you for taking time of your day to attend our session, and we look forward to seeing you at the future Scintica event. Have a wonderful day. Thank you.